Good evening, everyone. Today is your week 3 session for the course Structural Biology. So, in this session, we will be talking about the week 3 topics in which you have covered the introduction to structural biology techniques, in which you have looked into the X-ray crystallography and how it works, and what are the different aspects of X-ray crystallography, as well as how the crystallization is important for X-ray crystallography, as well as how to mount these crystals for getting better diffraction patterns. So in this, firstly, we'll summarize all these topics, then we will practice few MCQs. We will look into the current literature, and then I'll be taking your doubts. Before talking about the X-ray crystallography, in the last week, you have a doubt in the intrinsically disordered proteins. So this topic was not discussed in the previous week session. So in this week, we'll first look into these IDPs. So IDPs, or the proteins with the intrinsically disordered regions, they make up around 30% of the human proteome. And the main feature, or you can say the characteristic of these IDPs or IDR, is that they do not fold spontaneously into a single or well-defined 3D structure. For example, in this case, as you can see, it is a structured protein which is well folded. But if we talk about the intrinsically disordered proteins, then they are continuously moving. They're not getting completely folded into a proper well-defined structure. So that means it is fluctuating between multiple conformations. So this is referred to as intrinsically disordered protein, which is disordered. And you can also find certain proteins which are properly folded, but they have a region which is intrinsically disordered region. So that particular region is flexible, whereas the other part is properly folded or completely structured. And then the IDPs or IDRs, they cannot be readily categorized by the traditional methods which we use for the structural biology. So there is a need to develop new technologies to investigate their structures and the behaviors, that what is the relevance of these intrinsically disordered proteins. So we require new technologies. So now we'll talk about the characteristics of these IDPs. So the first characteristic which we have discussed is that they lack a stable secondary or tertiary structure. That means they will not adopt a proper alpha helix or beta sheet. So they just means you can say they fluctuate among various conformations. They are highly flexible and dynamic because they are not obtaining a constant structure. They are enriched in polar and charged amino acids, for example, glutamine, serine, and lysine. So generally, they are composed of these amino acids. And due to the nature of these amino acids, they remain flexible. They do not adopt a proper structure, depleted in hydrophobic residues. So in the previous session, when we were talking about different models of folding, then we have seen that how the hydrophobic interactions, they have important role in the protein folding. So these IDPs or these IDRs, they lack these hydrophobic residues. So which is also a reason that why they do not fold into a proper stable structure. So they lack these hydrophobic residues. And the last feature is that often functionally important despite structural disorder. So yeah, they do not have proper structure, but we cannot say that they do not have any function. So irrespective of structural features, they have some functional relevance. So now we will look into that what are the importance of these IDPs. Means even if they do not have well-defined structure, but still they have their role in function. So what are those functions? So the first function is regulation and signaling. So these IDPs, they can interact with multiple partners, which make them key players in cell signaling and regulation. So in this way, as they can interact with multiple partners, so they have a role in signaling and regulation. For example, there is a gene named as P53, which is a tumor suppressor protein. So it interacts with numerous regulatory proteins and it is involved in controlling the cell cycle as well as apoptosis. So apoptosis is the programmed cell death. So this particular protein, it decides that how to control the cell cycle, whether the cell should undergo continuous division or whether it should undergo apoptosis if there is any damage. So in this way, these IDPs, they have important role. They can decide, they can regulate, or they can control the behavior of a cell. Then the second feature is molecular recognition and binding. So their flexible nature allows them to bind to different targets with high specificity. So these proteins, if a protein is particularly or you can say if it is folded into a stable tertiary or secondary structure, for example, beta sheets or alpha helices, then there is a limitation. There is a specificity for a particular, you can say, particular substrate to which that particular protein can bind. But now, in the case of IDPs, as they have flexible nature, they do not have a stable or the constant structure. So now, due to this flexibility, they get the ability to interact with different targets along with the high specificity. So this is one of the advantage. And one such example is of c-MYC, which is a transcription factor which regulates the gene expression and it interacts with DNA and proteins. So now next feature is that post-translational modifications. So now these IDPs, they are also the site for phosphorylation, acetylation, and ubiquitination, which regulate their function. So in this way, they also allow for these modifications, and these modifications can further on decide that what function a protein will perform. So that's why these IDPs are also acting as a sites for these post-translational modifications. So example is tau protein, which is involved in microtubule stability. So it undergoes phosphorylation and which is linked to neurodegenerative diseases. So now you can see that how the phosphorylation is influencing this particular protein. So its phosphorylation is linked to neurodegenerative diseases. So in this way, they are providing the site for these modifications, and these modifications are ultimately bringing about the some functional consequence. So in this way, this is also one such importance of IDPs. Now next is of signal transduction. So they have critical role in cellular communication pathways. For example, CRAB, which is CAMP response element binding protein. So it is an intrinsically disordered protein which is involved in gene transcription regulation. So in this way, they are also involved in signal transduction processes as well. Next feature is adaptability in stress responses. So these IDPs, they also help in stress tolerance by interacting with chaperones and preventing aggregations. So due to their flexibility, they are also having the ability to interact with chaperones, and due to this flexibility, they do not get aggregated. So that's why they are involved in stress response as well. For example, heat shock protein 27. So it is involved in protein homeostasis under stress conditions. So under stress conditions, there is an ability that protein might get aggregated. But due to this IDP, they are now able to interact with chaperones, which allow them to properly get folded into a proper structure. And they also avoid the aggregation. So these are some of the importance that why the IDPs are important. And now we'll talk about certain examples. So these examples we have already discussed, such as P53, which is important in cell cycle and apoptosis. Then we also talked about the tau protein, which is important in neurodegenerative disorders. And we have seen that these tau proteins, so their amino acids which are present in this protein, they act as a site for phosphorylation or post-translation modifications. So these modifications, they are involved in the neurodegenerative disorders. Then there is example of alpha-synuclein, which is associated with Parkinson's disease. So this protein is also an example of IDP. Then BRCA1, or you can also call them as BRCA1, so which is majorly involved in breast cancer. So it plays a role in DNA repair and cancer suppression. Then E1A, which is adenovirus protein, which is also involved in transcription regulation. So in this way, we have seen that these IDP proteins, although they do not have well-defined structure, but they have some functional consequence on the cell, and they can determine that what will be the means how it will interact with other proteins and what functionality will that IDP will bring. Now, relevance in disease and drug development. So now we'll see that whether they have any importance for designing drugs. So now these IDPs, they are linked to disease such as cancer, neurodegeneration, and cardiovascular disorders. So in such cases, if we target these IDPs with small molecules or peptides, so it is an emerging strategy in drug discovery. So generally when we design drugs, so firstly we need to find certain proteins or the biomarkers which are involved in a disease. But as we have seen that there is development in the technology, now we are able to identify the IDPs which are responsible to certain disease. So now if we know those IDPs, we can design small molecules, or we can say we can design some drugs so that which can bind to these IDPs, and then they can control the expression of these IDPs so that we can design the drugs to treat that disease. So now these IDPs, they play essential role in cellular function, regulation and disease mechanisms. Their flexibility and ability to interact with multiple partners make them unique in biological systems, making them attractive targets for therapeutic interventions. So in this way, this is the whole summary that these IDPs, they have essential roles and their flexibility is providing them advantage for multiple partner interactions, and that's why they are also the attractive target for therapeutic interventions. So uh, Okay, so now, according to literature, if we look at the number of IDPs which have been published in PubMed versus the publication year, then we can see that with the passing year, the number of IDPs, they have increased. So their publication, that means their reported cases, they have increased. So this shows that how the technological advancements, they have helped us to identify more and more IDPs. So now we have a more knowledge about these IDPs. Now we can study their functional relevance as well as we can also design drugs against them. So now this is a difference between a structured domain versus a disordered region. So a sequence, it will have a proper structure, it will have some functions such as enzyme catalysis. An example is Tim barrel, which is a fully folded, stable folded structure. Whereas if we talk about the disordered region, then in this case, the example is HSP70, which is a heat shock protein 70, and which have a conformational ensemble. That means conformational ensemble means that it does not adopt a single stable conformation, but there is an ensemble that is there is a combination of different conformations which it can take. So it is a disordered monomer. So its sequence and it is disordered. So it do not have a proper structure, but still it has some function. For example, it is also involved in enzyme catalysis. So this shows that structure function paradigm which is established, whereas disordered function paradigm it is still emerging. So this shows that disordered regions they are not irrelevant. They are also having some functional significance. So now this particular paradigm is emerging. Now people are focusing more on these IDPs to understand their functional relevance. So this is all about the intrinsically disordered proteins. Now we will move to the week 3 content. So in week 3, you have looked into the X-ray crystallography, but before talking about X-ray crystallography, we will talk about the cellular organization, that how the different cells are organized in the body and how we can study them. So cellular functions, they rely on molecular communications. So there are various macromolecules which are present inside the cell, which constitutes the cells. So now as you can see that the atoms, which is the smallest unit, which combines to form a molecule, then these molecules, group of molecules combine to form a macromolecule, that means a bigger molecule. Then these macromolecules further make the organelles. So organelles are the small functional groups or small functional bodies which are present inside the cell, which perform certain functions. So now these cells, so group of cells, they combine together to form a tissue. Now, particular tissue, multiple tissues combine to form an organ, and different organs constitutes an organ system, and ultimately, different organ systems give rise to an organism. So, in this way, as you can see, that how the cellular organization is present. So to understand biological processes, we need to visualize the molecules at atomic levels. So to understand the proper functionality inside an organism, we need to move back to the smallest unit, which is the atom, and we need to gain the understanding of those small unit to understand what are the different biological processes which are undergoing or which are taking place. So structural biology techniques, they help us to determine the arrangement of atoms in a biological macromolecules. So if we can study them, then we will be able to visualize a clear picture that how the different functions are brought about inside the human body or inside an organism. So now what is the requirement for this structural biology? So to visualize these biological macromolecules, there is a condition that the wavelength of light must be smaller than an object. So if your object is smaller than the wavelength, then you won't be able to visualize it. So this is one of the conditions that you must have the light whose wavelength should be smaller than the object. So if we talk about the visible light, which lies in the range of 380 to 650, so or you can say 400 to 700 nanometers. So it is too large to image the proteins. So if we talk about proteins, then their atoms are about 1.5 angstrom or 0.15 nanometer apart. So we cannot visualize them with the visible light because this have the wavelength which is greater than the size of these particles. So now if we talk about X-rays, then they have the range of wavelengths from zero point 0.1 nanometer or you can say 1 angstrom. So it is used because their smaller wavelength allows the atomic level resolution. So this is the distribution of different lights. So these are the radio waves. And as we move towards the left side, so now we are moving towards the higher energy and short wavelengths. So what we require? We require that our light should be of shorter wavelength so that we can visualize the smaller objects or you can say smaller atoms. So to view at the atomic level, we should move to the left side from the in the electromagnetic spectra, we will be moving towards the left side. So from visible light, if we move to ultraviolet, it has the shorter wavelength, higher energy. Then to X-ray, which will also have high energy and shorter wavelength as compared to the visible light. And then the gamma rays. The X-rays becomes the candidate rays which could be used to visualize the means macromolecules at the atomic level. So in this way, that is the reason that why we have moved to X-ray crystallography and we cannot visualize these atomic level details with the microscopy, so with the visible light. Now we will see that the techniques can be divided into two categories. So one is the high resolution techniques and the other one is the low resolution techniques. So the high resolution techniques are the one in which you can distinguish among the objects clearly. So atomic level resolution is higher. That means you can clearly distinguish among the two atoms which are present inside the macromolecules, whereas low resolution means that you cannot properly distinguish among themselves. So the example of high resolution techniques are X-ray crystallography, or they are also referred to as protein crystallography in this sense. Then comes nuclear magnetic resonance or NMR. Then comes cryo-electron microscopy, which is cryo-EM. So these are the high resolution techniques. And in this case, we have shown that what is the size range of the object which is investigated. So towards the left, the size is decreasing. So the best is the NMRs, then X-ray, then cryo-EM, and ultimately the light microscopy. So light microscopy do not allow us to visualize the molecules at the higher resolution. But then if we talk about cryo-electron tomography or single particle cryo-EM, then they are better. And then X-ray and NMR, they are much better than these cryo-EM microscopy techniques. So in the next, if we talk about the low resolution techniques, then the examples are circular dichroism, fluorescence spectroscopy, electron paramagnetic resonance, and small angle X-ray diffraction, which is also known as SAX. So which is small angle X-ray diffraction method. So these are the low resolution techniques. So for the today's session, our main interest is the X-ray crystallography. So if we talk about the basic concepts of X-ray crystallography, then the X-rays are the form of electromagnetic radiations with higher energy and shorter wavelength. The problem with X-ray is that a small molecule weakly scatters the X-rays, so making it difficult to detect. So if we have a single molecule, then it is difficult to view by the X-ray crystallography. So solution is that instead of using a single molecule, we use the crystals. So what are crystals? So crystals are the 3D array of molecules, and the molecules could be 10 raised to power 14 molecules that reinforce the diffraction signals. So these molecules, you can see that the smallest unit is known as unit cell. When they are arranged properly into a 3D array or 3D group, then it is a crystal lattice. So now if you have multiple molecules, then the ability of X-ray to scatter them or to generate a diffraction pattern increases. So that is one of the solution, that instead of using a single molecule, we go for the crystal formation and then observing the X-ray diffraction pattern by using a protein crystal. So now what are the key challenges? So X-rays cannot be focused using the lenses, unlike visible light. So in visible light, we use the lenses to focus the light on that particular specimen and then we visualize it with the help of that lens. But in case of X-rays, we cannot use it. So instead, we use the computers to simulate an image by reconstructing the diffraction patterns. So now whenever the X-ray will strike an atom, it will get refracted and then that particular pattern will be recorded by the computers and then it will help us to reconstruct the image. This is one of the device which is used for observing the patterns. So it has the X-ray source which will pass the X-ray through a specimen. Then there is a goniometer on the top of which there will be a specimen or the crystal of protein for which we want to visualize the structure. And then it will rotate into different directions and it will allow us to obtain different angles of diffraction. So ultimately this detector, it will be able to capture those different diffraction patterns and it will help us to visualize the image. So in this way, this is one of the difference among the visible light microscopy versus X-ray crystallography. Yeah, so this is one of the representation of X-ray crystallography. So we have a X-ray source, then we have a material of interest. So these are the crystals. And then if we pass the X-ray, then there could be wide angle scattering as well as small angle scattering. So as we have discussed in the previous slide, that one is the X-ray crystallography and another one is the small angle X-ray diffraction pattern. So small angle X-ray scattering, which is known as SAX, so which is a low resolution technique. So in this case, it provides us the information about the structural arrangement, that how the atoms are arranged in the macromolecule, or you can say in the protein, how those atoms are arranged as the basic unit in the materials. And it provides us the information about the size and shape of the materials. So in the later slides, you will be getting the clearer picture that how the X-ray crystallography helps you to visualize the protein structures. So if we talk about the little bit about the history of X-ray crystallography, then it is the main method which is used to determine the 3D structure of macromolecules. And the first protein diffraction pattern was obtained for the pepsin, so which is an enzyme. So there were two scientists. So one was John Desmond Bernal and another was Dorothy Hodgkin. So these two people, they got Nobel Prize in 1934 for this diffraction pattern of the pepsin. So advantages of this technique is that there is no size limit for macromolecule analysis. So there is no such limit. You can observe of any sized macromolecule, even if it's bigger, smaller, at the atomic level resolution. But the limitation of this technique is that it requires high quality crystals and it does not capture the dynamic states. So in this way, if you are not able to crystallize the protein, then you won't be able to visualize it. So this is one of the limitation. And the another limitation was that it is capturing a particular conformation at a stable state, or you can say the constant state. So it cannot capture the structure at dynamic states. So there might be certain proteins which adopt different structures. So in that case, it will be a bit of a problem to means observe its structure using X-ray crystallography, because you will be only observing a single state at a time. So then there is also another challenge in X-ray crystallography, which is a phase problem. So X-ray diffraction patterns, they give you information about the amplitudes, or you can say the intensity of diffraction, but not the phases of diffraction. So solving the phase problem is essential to reconstruct the electron density of a crystal. So in this way, you need to solve this problem so that you can get the electron density map of a molecule, and ultimately you can design the structure, or you can means know the structure, that what is the structure of that particular macromolecule. So in this way, we need to solve the phase problem. And in the coming weeks, we will be looking into the details of the phase problem, that how it is tackled and how it arises. So now we will talk about the steps which are involved in the protein crystallography. Sorry for the network. There was a network issue. So, okay. So now we'll talk about the steps in the protein crystallography. So there are these steps. So first step is target protein identification, in which you have to select a protein of interest. So first of all, we need a protein which we want to visualize. So now first step is to select that particular protein. Second step is cloning and overexpression. Now you have obtained the protein, but you should make sure that you have the enough or sufficient quantity of that protein. So for that purposes, you need to perform cloning of that protein. You have to means undergo PCR so that you can increase the concentration of that particular protein for which you want to analyze the structure, or you have to overexpress it. Then the third step is purification, which is also very important, because if you want to make the crystals, then your protein should be very pure. So isolating the protein for crystallization. Then the fourth step is crystallization. Now, as you have obtained the sufficient quantity of a protein, you have purified it. Now you want to generate the crystals. You want to convert the protein into crystals for X-ray diffraction. Now, after the crystals have been generated, next step is crystal mounting and data collection. So now how you mount these crystals, it is also very important. So you have to expose these crystals to the X-rays. So for that, they should be properly mounted. Then comes phase determination, in which you have to resolve the phase problem so that you can get the electron density map. So in this case, we will be resolving the phase problem, and then we will be able to get the electron density map. This electron density map will help us to construct the atomic structures. So in the next step, which is the model building and refinement. So we refine the structure, and then ultimately when the structure is completely refined, model is built. So last step is to submit the data. So we will deposit it in the database, and one such database for the protein structure is PDB, Protein Data Bank. So these are the overview of the steps involved in protein crystallography. Now one by one we will be looking into the detail of these steps. So first step is finding, optimizing and preparing the target gene. So for gene identification, we need to find the gene of interest. So because these genes are ultimately forming the proteins by translation. So what we will do, we will take the gene for which the for means which give rise to a protein for which we want to analyze the structure. So firstly we will identify that gene, and then we will design the primers and let it undergo PCR amplification so that number of copy of that gene can be increased. So for that, we have the gene of interest. There will be some restriction sites. So restriction sites are the regions which are detected by the restriction enzymes, and restriction enzymes are very specific. They detect these sites and make a cut in the, that means the gene of interest. They will make a cut on the site where they recognize the sequence. So with the restriction site, they will make some cut, and this sequence which is present in the restriction site, it will be very specific to a particular restriction enzyme. So on both sides, it will generate cuts, and it might generate sticky ends. So in this case, we need sticky ends so that we can ligate them to form a circular plasmid. Because in blunt ends, it's very difficult to make them circularize. So in this way, we will select the restriction enzyme which make the sticky ends, or you can say the staggered cuts, in which we observe the overhangs. So similarly, we will take a bacterial plasmid. So these plasmids, what we do is that we take E. coli as a host, we take gene of interest. Plasmid are the extra chromosomal materials present in the E. coli. So E. coli is acting as a host. It has the extra chromosomal material which is plasmid. We will insert our gene of interest into the plasmid. This plasmid will be entered into E. coli. Now this plasmid will divide as the E. coli will divide. So as we know that the E. coli has the faster doubling rate. So that's why we use the E. coli, so that it can generate multiple copies of our gene of interest, because it is doubling, and it is also means increasing the number of plasmids. So if we have gene of interest inside the plasmid, then the chances of doubling that gene of interest will also increase. So that is one of the reason that why we select E. coli as a host. So similarly, in this bacterial plasmid also, we will digest it with the similar restriction enzyme, so that the sticky ends at the sequence at the sticky end remains constant in both these gene of interest as well as bacterial plasmid, due to which now it will be able to get circularized. So these sequences, there will be similarity, they will be complementary to this particular sequence, and this end will be complementary to the this particular end. So that's why they will get bind to each other, they will get circularized. So the main key player is the restriction enzyme and it should be same. So here is an example that how a restriction enzyme works. So for example, I'm taking the example of EcoR1. So it has a restriction site which is composed of 6 nucleotides. So this is the particular sequence. So it cleaves at this particular site. Now the sequence which it will generate will be like this. And you imagine that you have the another plasmid in which you will be generating the ends in the same way. So in that also these two ends will be generated. So now what will happen that instead of the right side of the gene aligning or means annealing to the left part, the another gene, which in this case is the bacterial plasmid, so it will get annealed to the chopped or you can say the restrict means digested fragment of the gene of interest. So in this way, ligation happens. So DNA ligase will come, which will ligate these, and there will be bond formation between G and A and G. This cut will be sealed. So in this way, the DNA ligase will seal the gap, and similarly, the plasmid will get circularized again with the gene of interest. So now the thing which is formed is the recombinant DNA because it contains the gene of interest, so it is recombined. Now we'll talk a little bit about the vector because vector, the plasmid is a vector. So vector is like a vehicle which is carrying the gene of interest. So vector is a biological tool in rDNA technology. So rDNA is nothing but it is a recombinant DNA technology and it is used for the delivery of desired foreign DNA into the host cell. So in this scenario, our foreign DNA is the gene of interest and host cell is the E. coli in which we want to express that gene of interest. So vector is a DNA molecule that have the ability to replicate autonomously in the host cell and into which the DNA fragment to be cloned. So these are the features of the vector, that a vector should have ability to replicate and it should also have ability so that it can means we can integrate the gene of interest into it. So now there are certain properties of ideal vector. So one such property is that it should be replicate autonomously, that so that it can replicate and the gene of interest can also get doubled or tripled. So that gene will also get replicated multiple times. So we want to increase the number of gene of interest. So that's why this is one of the property that it should be able to replicate autonomously. Second thing is that a vector should be less than 10 kb in size. So why this so? Because if it will be smaller in size, then it will be better able to take the gene of interest. If its size will be large, then it is difficult to integrate the gene of interest into that vector. So its size should be less than 10 kb. Then it should be easily isolated and purify. So for example, if we are not able to isolate it, then how come we will insert the gene of interest into it? So this is also one of the important property that it should be easily isolated as well as purified. Then the next thing is that it should be easily introduced into the host cell. So now if we convert that particular vector into a recombinant molecule or you can say recombinant DNA, then it should have the ability so that it can easily get introduced into the host cell. Host cell should not reject it because our ultimate aim is to allow it to replicate. So it should be easily introduced. Next thing is that it should have suitable marker genes. So now if whenever we construct a recombinant DNA, then we want to distinguish it from the another means dividing another material or another, there is a possibility that there is another plasmid which is present in the that particular E. coli because when we isolate those plasmids after the means after performing the rounds of replication, then we will be isolating those plasmids and obtaining our gene of interest. So in such case, where we want to distinguish that whether the plasmid contains our gene of interest or not, so marker genes are very important because they will mark the plasmid. So that we can distinguish that whether it is a recombinant plasmid or whether it is a normal plasmid which do not have our gene of interest. So that is one of the property. Now vector should consist of a unique target site and a recognition site for the restriction enzymes. So as we have seen previously that what we do is for the gene of interest, we cleave it with the restriction enzyme and same restriction enzyme is used to cleave the vector. So if it does not have the same recognition site, then we won't be able to cleave both of them with the same restriction enzyme. And then they will not be able to circularize because the cuts which are produced or the sequence which we obtain, they will be different. So to avoid that, we want that there should be a unique target site and there should be a recognition sites for various restriction enzymes so that those enzymes can be used to cut both the gene of interest as well as the vector. Next feature is that it should have the capability to incorporate either itself or the DNA insert into the genome of the host. So this is also very important because if it will not insert the gene of interest means into the host cell, then the means ultimate aim of the cloning will fail. We want it to clone the gene of interest. So this is also one of the important feature that it should be able to incorporate itself into the genome of the host cell. So this is one of the example of the vector. So in this case, for simplicity, we will not go into the details. So I have taken the example of the plasmid vector, which is present in the E. coli as well as different bacteria. So the PBR322, it is the name of that particular plasmid vector, and this naming is based on the, so P means plasmid and B means Bolivar Rodriguez. So these are the scientists who have identified it. And 322 is just a serial number to distinguish that particular plasmid. So there is a series of plasmids which might have been identified. So to distinguish among them, there is a serial number which is given. So this particular plasmid, it contains two set of antibiotic resistance. So for example, ampicillin and tetracycline. So ampicillin and tetracycline, these are the antibiotic resistance, and they are very important because whenever you will insert your gene of interest into this plasmid, then there are certain sites where we will be inserting it. So for example, as you can see, BAM-H1, SAL1, EcoRV, these blue colored things which are written, these are the sites of the restriction enzymes where the restriction enzymes are cleaving. So what we do is that we cleave this particular plasmid at these sites, means we will use one such restriction enzyme. For example, if we are using BAM-H1 to cleave this plasmid, same enzyme is used to cleave the gene of interest. Now, when that particular gene of interest will get inserted into this region, so what it will do? It will break the site or break the sequence of this tetracycline resistant gene. So now when we will provide a medium where tetracycline is given antibiotic, then the plasmids will die off, which contain the recombinant DNA, which contain the gene of interest. So in that way, this particular thing is important so that we can distinguish that whether our plasmid is means it has taken up the gene of interest or not. But as you can see that in this case, um, means it is not helpful because if you will insert your gene of interest into this and if you provide tetracycline, so in that way if tetracycline sequence is disturbed, so it will no longer be able to survive in that medium, so it will die off. But we do not want it to die, we want it to replicate more. So in such cases we avoid the insertion, or we avoid to distinguish it using tetracycline. Instead we will use some another antibiotic, or we can use some other marker regions. So in that way, we require certain marker genes which are not antibiotic resistant, but some other genes, so that we can distinguish and we do not have to lose the plasmid in order to identify. Okay, so it contains only single or unique recognition site for 12 different restriction enzymes. So in this case, we have also discussed that for an ideal vector, it should also have different sites for different restriction enzymes. So in this case, as we can see, there are multiple sites and there are 12 different restriction enzyme sites. So that's why it is helpful that we can use such restriction enzymes and we can insert our gene of interest into it. So these are some of the examples, and I won't go into the detail of different types of vectors which are available, but yeah, there are different vectors which are already available, which have the better marker genes, which help us to distinguish. Instead of disturbing the tetracycline resistance or antibiotic resistant part, we use the other strategies. For example, there are certain vectors which have the ability, they interact with some chemicals and they generate different colors. So in that way, to distinguish among different colored vectors is very helpful, and they won't disturb the gene of interest or the plasmid means or the vector. Okay, so next step is the protein purification step, in which we generally use the chromatographic techniques. So till now, what we have seen so far is that firstly we need the protein of interest. So for that, we first go to identify the gene which is giving rise to that protein. We obtain that gene, we clone it, and increase the number of such copies of the genes and express them using the plasmids. So in the bacteria, multiple number of copies of genes will be present, they will be expressed. So more number of protein will be obtained. So now if greater concentration of protein is obtained, our next step is to purify that protein so that we can go for the crystallization step. So in purification, we use different chromatographic techniques. So one such chromatography technique is gel filtration chromatography, and it is also known as the size exclusion chromatography, because in this case, the separation of different molecules, they are based on the size. So other names are gel permeation chromatography as well. So as you can see in this figure, so what we do that we have this particular column in which we have the porous polymer beads. So these beads, they have some small pores. So as soon you enter the protein mixture into this column, so now what will happen is that these smaller molecules, which are smaller means protein molecules which are present, they will get attached into this pores. They will get bind with the, they will enter into the pores and they will get trapped in these beads. So in such case, the molecules, or you can say the proteins which are larger in size, they will elute out first because they won't be able to enter into these pores. So now they are simply passing down the this beads. So what happened that very small molecules, they enter many pores in the gels which equilibrate between the gel and removing the buffer, and so travel slowly and eluted later. Whereas the medium sized molecules, they will enter some of the pores in the gel which will equilibrate between the gel and the moving buffer. Whereas if we talk about the large molecules, then they will enter few pores, or means you can say negligible pores, and they will travel rapidly and will be eluted sooner. So in this way, the molecules which are larger in size, they will get eluted first, and the molecules which are small in size, they will elute later. So in this way, you will be able to separate or purify the proteins on the basis of their sizes. So this is one of the technique. Now, next such chromatography technique is the ion exchange chromatography. So in this case, if you want to separate the molecules based on their charge, for example, you have different protein mixture. Now you want to separate these proteins on the basis of charge. So you will go for the ion exchange chromatography, and it is a common method which is generally used. So depending on your protein of interest, for example, if you want to separate the protein which is negatively charged or positively charged, so you will use the surface which will be of the negative charge to the protein of interest. So in this case, if we want the to means purify the negatively charged protein, so what we will do, we will take the surface which will be the cationic in nature, which will have a positive charge. So whenever you will flow the solution, which is the protein mixture, whenever you will flow the protein mixture, so what will happen, that the negatively charged proteins, they will get binded to the surface because our surface is positively charged, whereas the positively charged proteins, they will flow out because of the repulsions, they will not be able to interact. So in that way, all the positively charged molecules will get eluted first, and with time, in the end, we will add the further elution buffer. So elution buffer is a liquid which help us to elute the protein mixture. So it helps the protein mixture to flow from this particular column. So now in the end, we will add the elution buffer to separate the slightly negatively charged molecules. So now the molecules which are slightly charged, firstly they will elute out, and ultimately the protein molecules which are means means strongly negative, which are means highly negatively charged, they will elute later. So this is a way that how you can separate the proteins on the basis of charge, and similar thing can be reversed as well. For example, if you want to means instead of obtaining the negative proteins in the end, you if you want the positive proteins in the end, so in place of cationic surface, you will use the anionic surface. So in that way, there are different types of such surfaces which exist. So there are some cationic exchangers, some anionic exchangers. So depending on the charge, this help to purify the proteins. Then the third method of chromatography is the affinity chromatography. So as the name suggests, affinity. Affinity means there is some kind of binding, or means something binding is happening. So that is known as affinity. So in this case, there is a specific binding, and one such example is of the his tagged proteins using the nickel affinity chromatography. So what happened here is that you take the column in which you have the surface where you have some chelator. So in this case, we generally use NTA as a chelator, which bind with the nickel. So in this case, in the surface, we will be having the NTA and nickel, which is present. And as soon as you will flow the protein mixture, so what will happen? The proteins which are tagged with histidine, they will bind with this nickel because there will be some kind of interactions between the nickel and histidine. So in histidine, we have a imidazole ring. So the nitrogen present in the imidazole ring is very important to interact with the nickel. So there will be a coordinate bond formation which will take place. So now what will happen is that the proteins which do not have the histidine tags, they will flow directly. They will elute out first. And whereas the proteins which contain the histidine tag, they will get bind to these nickel tags or nickel NTA tags, and they will remain in the column. So ultimately in the end, what you will do, you will provide the elution buffer along with the imidazole. So imidazole, what it will do, as soon as you provide imidazole, it will replace. So instead of histidine binding to nickel, it will compete with the histidine and imidazole will replace the histidine. So now what will happen? The proteins which were previously tagged with histidine, now they will move out and imidazole will take their place. So this is the representation that how it happens. So before adding imidazole, proteins which we want to study, it has the histidine tag. Now these imidazole rings which are present, so their nitrogen which is present, it is interacting with the nickel ion. So these proteins will get tagged or attached to this beads. They will not move out, they will not get eluted. And in the second phase, when we want to obtain these, because after all the proteins get elute out, and now only the histone tag proteins are present, sorry, histidine tag proteins are present. So what we will do, we will add the elution buffer, and along with it, we will add the imidazole. So now imidazole will replace the histidine tagged proteins and it will get bind to nickel, and now these histidine tagged proteins are free, so they will get elute out. So this is the way that how we can elute out the protein of interest. We can purify it on the basis of the tag which is applied on it. And now the fourth step is protein crystallization. So till now we have seen that how we have obtained the sufficient quantity of the protein, we have purified it. Now after all these steps, the next step is crystallization, which is very important to observe the protein with the help of X-rays. So now we want to crystallize it. So for that, protein must be highly concentrated because we cannot work with the small quantity of protein. So it should be concentrated, it should be homogeneous, and it should be stable. So for the purification, because we want to obtain the homogeneous protein, that's why we are purifying it. We do not want contamination of multiple proteins in the single crystal. So it should be homogeneous. Then crystallization involves forming a regular 3D array of protein molecules. So regular 3D array of protein molecules, that means crystal. So that is a proper organized or regular or ordered structure. So we want that ordered structure. And it was first observed in 1840 and in which hemoglobin crystallization was performed. So, okay, I'll just Okay, so firstly, the protein crystallization was observed in 1840 for the hemoglobin crystallization, and then James Sumner in 1926 he crystallized the first enzyme which was urease, and then Bernal and Hodgkin in 1934 they demonstrated that the wet protein crystals produce better diffraction patterns. So these are some of the historical things about the protein crystallization. And we have also discussed that Bernal and Hodgkin were the one in 1934. They observed the X-ray diffraction pattern for the pepsin enzyme. So X-ray diffraction and structural determination is the last step in which the crystals are exposed to X-rays, their diffraction patterns are recorded, and ultimately this data is processed using various computational methods to reconstruct the 3D atomic structures. So in the later sessions, we will go into the detail that how different things are done. Means it is not just these two steps, there are multiple steps which are involved, but our main topic of interest here is the crystallization. So now if we talk about protein crystallization, it is a crucial step which helps to determine the 3D structure of the biological macromolecules. Now, what are the goals of crystallization? So, first goal is to obtain the high quality protein crystals for structure determination. Second aim is to understand the crystal growth mechanisms and ultimately to develop the techniques for optimizing the crystal formation. Because if we have proper crystals in our hand, then we will be able to observe them using X-ray crystallography. So this crystallization is the major step which is responsible for proper X-ray diffraction patterns. And it is very hard, and still now we do not have well defined rules that how we can crystallize a protein, how we can obtain the high quality protein. So that's why it is very important to understand that how we can generate a well high quality crystals. So these are some of the examples of different crystals, protein crystals which are formed. So before moving further, we must talk about that how the protein structures or protein crystals vary from other crystals. What are the unique property of these crystals? So macromolecules such as proteins, DNA and RNA, they can form crystals. So what are crystals? Crystals are the structures which are held together by the non-covalent interactions. So in this, there are some non-covalent interaction between those molecules which give rise to the crystals. So protein crystals are unique. They contain 30 to 80% of the solvent. So due to presence of this solvent in them, which make them fragile. So they are very fragile. And if you prick them, they will simply break. So they are fragile due to the presence of the solvent. Then allow the structural flexibility, enabling enzymes to remain active in crystals. So inside crystals, there is a structural flexibility for the interactions. So there are certain enzymes which can interact with the substrate and generate the product. So in that way, these crystals, they're not completely constant, but they allow for a little bit of the structural flexibility and then can accommodate small or large conformational changes without breaking. So in this way, these crystals, they allow for some of the conformational changes without even breaking. So these are some of the unique features of the protein crystals. And in this, I have shown the how these protein crystals appear. In this case, under the visible light, how the protein crystals appear, how the protein precipitate appear, and how the salt crystals appear. So in the later slides, we'll see that how we can distinguish that whether the crystals which we have obtained is the protein crystal or whether it is a salt crystal. So we will see in the later slide. Now we will talk about some of the key concepts in the protein crystallization. So first thing is supersaturation and crystallization zones. So means how we can get the protein crystallization, where are the means what are the regions where the crystallization is obtained. So for that we must be aware of the supersaturation and crystallization zones. So supersaturation is a thermodynamically unstable state where more solute is dissolved than usual. So as we all know that there is a saturation point where the amount of solute is maximum above which if we add the solute, it will not take it. But in the case where where there is a more solute is dissolved than usual. So when we cross that saturation barrier, that means we have reached the phase which is supersaturation phase. So this is a stable zone, you can say. So this y-axis represents the protein concentration, x-axis represents the precipitant concentration. So whenever you have a stable zone, in this case, the amount of solute is lesser in the solvent. So this is the under saturation when it has not reached the stage of saturation where we cannot add further solute. So this is known as stable zone or under saturation zone. And then our aim is to enter into this supersaturation zone where the solute is slightly higher than the usual solute which is required for saturation. That's why it is known as supersaturation. So it is achieved by vapor diffusion or the slow evaporation technique. So there are different techniques which are being used to enter into the supersaturation level. And supersaturation level, there are also further three zones. So one is the metastable zone, another one is the stable zone. In metastable zone also we have two zones. One is crystal growth zone, another one is nucleation zone. So in the nucleation zone, these smaller soluble means protein molecules, they will get aggregated. But in this case, they're not completely aggregated, so they enter into the crystal growth zone and they generate a crystal. But they might, if they cross this instability line, so this will be a state where there will be, where the solute or the protein concentration is very high, such that those proteins, they get precipitated, they get aggregated, and in this zone they will not be able to form the crystal. So in this way, I have divided crystallization zones into three main zones. So one is the metastable zone. So these zones are the under the supersaturation region. So one is metastable zone, in which no immediate nucleation is happening, but crystal growth can occur with a seed. So if you have a small crystal which is already available, which is not completely formed, so which is referred to as a seed. So in this case, there is no immediate nucleation, but still crystal growth can happen. Then nucleation zone is the region where the crystal begins to form. So in nucleation zone, these proteins, they will come together, they will aggregate, but not excessively. So a little bit of combining, a little bit of aggregation is happening, which can give rise to the protein crystals. Then comes precipitation zone, which is the highly unstable zone, in which excess protein aggregates rather than crystallizing. So when the number of aggregate of protein increases, then they will get precipitated and they will not be able to form the crystals. So in this way, these are three types of zones which are formed. Now there are multiple challenges which someone face when they crystallize the proteins. So one such problem is the nucleation problems. So nucleation problem means if you have too many nuclei, that means you have many tiny crystals. So instead of obtaining one of the large crystals, you have multiple such tiny crystals. So in such case, that is also the nucleation problem, in which you won't be able to get a single proper crystal which is large. Then no spontaneous nucleation. So in this case, crystals only grow with the seed crystals. So in this way, you need to provide some seed, some already available smaller crystal which is not completely formed. But if you do not provide it, then there will be no spontaneous nucleation. So that is also one of the problem. Then comes growth defects and solution conditions. So crystallization also stops due to defects or when the solution reaches the equilibrium. So if your solution reaches the equilibrium, then as we have talked that there are different methods to generate the crystals. So in such conditions, crystallization will stop. The crystals won't be formed if the equilibrium is reached. Second reason is that the surrounding solution, which is known as the mother liquor in which everything is happening, so it may differ from the original crystallization mix. So due to which the means this crystallization process might get stopped. So these are the reasons, or you can say these are the challenges which one faced while protein crystallization. So we need to keep these points in mind so that we can obtain better crystals. So now as we are talking about crystals, so we will also talk about the unit cell concept, because unit cells, they are the smallest repeating units which are present in the crystal. So multiple unit cells, they will combine and they will give rise to a proper crystal lattice. So in this way, it represents the unit cell. It is defined by three different axes, x-axis, y-axis and z-axis, or you can also call them as A, B and C axis, and each axis will have some angle. So in this case, angle between A and C axis is beta, angle between A and B axis is gamma, and angle between B and C axis is alpha. So in this way, unit cell have three different angles. It has three different axes, and repeating unit cells, it generates a full 3D crystal structure. So unit cells will be the smaller unit of a crystal. Now there are different factors which affect the protein crystallization. So previously we have seen that how the protein crystals are formed. Now there are certain factors which affect the protein crystallization. So one such factor is protein purity and concentration. For example, if you do not have a pure protein, then it will also influence the crystallization process. Your protein will not be able to properly crystallize because we need a homogeneous protein mixture to allow it to form the proper crystal. So that is one of the reasons. Second thing is concentration. So if you have, it means insufficient concentration of the protein for crystallization, then also you will not be able to obtain proper crystals. So proper concentration is also very much important. Then comes precipitating agents. So there are some precipitating agents which control the nucleation. So as we have seen that in nucleation, not aggressive aggregation is happening. There is a little bit of aggregation which is happening such that crystals can be formed. So in this case, if you are using certain precipitating agents, so if their concentration is high, then it might interfere with nucleation. So in that case, it might enter into the precipitation zone, which is the unstable, in which crystallization won't happen. So you need to make sure that the precipitating agents which are used, they should be in the proper concentration for to manage the proper crystallization. Then comes temperature and pH. So temperature and pH can also influence your crystal formation. If you have the imbalance of pH, or if the temperature conditions are very high or too low, then also the proper protein crystals won't get formed. Then there are certain additives such as detergents, cofactors, or reducing agents which help to stabilize the proteins. So they will also interfere with protein crystallization. It depends that at what concentration you are using these additives. So there should be a proper balance of all these things. So they will determine that whether you will be able to obtain proper crystals or not. So these are also the challenges, because we do not have a proper guideline or something. You can say we do not have a standard concentration which we could use and obtain the proper crystals. So that's why many people call it as a hit and trial method. So they use some personalized strategies, personalized concentrations, and they are able to obtain proper crystals. So in this, I have given us a short table. So different factors. So these things are again mentioned in these cases. So they are a bigger list of factors which affect the protein crystallization. Now there is a consortium, which is the Structural Genomics Research Consortium, which has the goal to determine the 3D structures of the health-related and biotechnologically important proteins. Um, Okay, so this particular consortium, it is a very important. It is responsible to determine the 3D structure of health-related and biotechnologically important proteins, because if as we have seen that how crystallization is difficult to achieve. So there are multiple challenges which are there. So this consortium came up and it decided that we will take the proteins which are involved in some health related or biotechnologically which are important. So we will select those proteins and we will firstly we will determine their 3D structures. Instead of performing the structure analysis of a bigger pool of protein, they are focused on some specific category of proteins. So part of the protein structure initiative of NIH, which is National Institute of Health, USA, and they works on the open science principles, avoiding patents, so that everybody can use those structures. So they do not focus on patents and all those things. And there are key centers of the Structural Genomic Research Consortium. So these are the New York Structural Genomic Research Center, Northeast Structural Genomics Center, Midwest Center for Structural Genomics, and Joint Center for Structural Genomics. So these are some of the key centers. And then SGRC research workflow. So this is their workflow, how they work. So they perform gene cloning and expression. So these will be the same steps which we have already covered. So first step is gene cloning and expression, then protein purification and crystallization, then X-ray diffraction and data collection, and ultimately structure determination and its functional analysis. And then after all these workflow, after all these steps being performed, the data which is obtained, they will upload it on the means um, available or publicly available databases, so that everybody can access that data, because they are focusing on such health related proteins. So if we properly understand their function and structure, then only we will be able to design drugs against them and understand them, that how we can cure for that particular disease. So in that way, this particular consortium is playing very important role towards the health industry. Now, what is the importance of growing protein crystals? So, why means this protein crystal is very important? So, single protein which is not able to diffract X-ray strongly, that's why we need crystals. So, crystals contain trillions of molecules which will help to amplify the signals. So, in this case, these are different unit cells. So, these individual horses represent a single unit cell. So, if we have multiple such molecules, then the ability to obtain the signals will be higher. So there will be amplification of the signal if we have trillion of molecules in the crystal. And ordered molecules produce strong diffraction due to constructive interference. So this constructive interference will only be achieved if we have ordered molecules and multiple molecules, not just a single molecule. So this is one of the reasons that why we need crystals for X-ray crystallography and why we are not directly working with the protein. Now, disorder in the crystals can affect diffraction. So, we need crystals, and along with this, we need the ordered crystals. So, if they will be in perfect order, then we will obtain strong diffraction pattern. So, here you can see that these single dots they represents the atomic positions, and they will means it represents the diffraction pattern. So, in this case, we obtain strong diffraction. We will see we are able to see a clear dots. Whereas if there is rotational disorder, so in this case, you can see that how these different layers of unit cell, how they are, means there is a rotational disorder. They are rotated to some angle. So they are not properly aligned. They are not in the order. There is some rotation which has undergone in different layers. So if there is such rotational disorder, then we obtain weaker diffraction. So in this case, as you can see, the diffraction pattern is very weak. You're not clearly able to distinguish different atomic positions. So this is referred to as rotational disorder. Another such disorder is translational disorder, which is also known as fuzzy, or which means give rise to fuzzy and streaky diffraction. So this is translational disorder. As you can see, it has a little bit of they have means each layer has undergone a little bit of translation. Not means in this they have rotated, whereas in this they are little translated. So in this case, the fuzzy or streaky diffraction pattern is observed. So this pattern is also not clear. So this slide shows that yes, we need protein crystals for X-ray crystallography. We cannot directly work with single proteins. And other than this, it also tells that we need crystals which should be in proper order to obtain the strong diffraction pattern so that we can generate the atomic resolution picture of that particular protein. So this shows the importance that why we need the protein crystals. Now we will talk about the little bit about the thermodynamics of the crystal growth. That how the crystal growth is happening, what is the thermodynamics which is involved. So for thermodynamics, we look into the free energy landscape. So in this case, free energy change for crystal is given by this formula. So delta G crystal is equals to delta H crystal. So delta H is change in enthalpy minus T delta S. So in this case, delta S is entropy. So entropy change in protein plus entropy change in the solvent. So if this particular delta G crystal is negative, which means that the crystal growth is spontaneous, it is happening. If it is positive, which means the crystal growth is unfavorable. So now we will look into the individual values. So firstly, we will talk about delta H crystal, which is the enthalpy change. Now our aim is to see that whether this thermodynamics of crystal growth, whether it is favorable or non-favorable. So delta H crystal, which is enthalpy change, so it looks into the formation of lattice contacts. So if lattice contacts are more, then it will be favorable. So in this case of crystal growth, if we talk about protein in crystal, then there are no such lattice contacts because each individual protein units, they are present far away from each other. Whereas if we talk about protein structure, or you can say protein crystal, then each individual protein unit, they are present close to each other. So there is a possibility of contacts, or you can say lattice contacts. So in this way, you can see that there are multiple lattice contacts which are observed in the protein crystal, which were not there in the protein solution. So that's why it has large area, there are specific contacts, and it will be rigid because molecules are present close to each other. So in this way, the delta H crystal or the enthalpy change is quite negative, that is from 0 to minus 17 kilocalories per mole. So if it is negative, that means it is favorable. So in this case, delta H crystal is favorable. Although this value is small, but it is favorable. Now the second term is T delta S of protein, which is the entropy change with respect to protein. Now we will see. So entropy means disorder, which means noise, that how freely the protein units, or you can say the protein units are moving. So in this case, it is unfavorable because you can see when protein is available in the solution, when it is present in solution, then each protein unit, it it has gaps in between, so it can freely move. There is no such limitation for its movement. So 3 degrees of freedom in orientation as well as 3 degrees of freedom in translation. So in this case, it is free to move. So in this case, the entropy of protein in solution will be higher. But if we talk about protein crystal, so as you can see that these individual protein units, they are tightly packed, there are lattice contacts. So now they are not free to move. So 0 degree of freedom in orientation and 0 degree of freedom in translation. So due to this loss of degree of freedom upon crystallization, there is drop in entropy. So this entropy change is low and it is from plus 7 to plus 25 kilocalories per mole, which is positive. So that's why it is unfavorable. So change in disorder is not high, which was higher in protein in solution. So this term is highly positive, which means it is highly unfavorable. Now the last term, which is T delta S of solvent. This is also the entropy change, but of the solvent. So previously we looked at the movement of protein. Now we will look at the movement of the solvent, that how freely a solvent can move. So this is highly favorable in the protein crystal. So when protein is in solution, then the water molecules are attached to this each protein individual unit. So now proteins are moving, not the water molecules are individually moving. So solvent movement is lesser and protein movement is more. Whereas in case of protein crystal, because each protein units are linked tightly, so now this water molecules, they are free to move. So their movement will be higher. There will be 3 degrees of freedom in orientation and 3 degrees of freedom in translation for these water molecules. So now entropy of solvent in protein crystal is high. So it is highly favorable and its value ranges from minus 7.5 to minus 50 kilocalories per mole, which states that water molecules are released upon crystallization. So this shows that the entropy change, that is T delta S of solvent, is quite high, which is highly favorable. So now we'll combine all these terms and we'll see that whether delta G of crystal is favorable or non-favorable. So if we combine all of these terms, then delta H of crystal, which was enthalpy change, it was favorable, which ranged from 10 to minus 17 kilocalories per mole. Then the second term, which was T delta S of protein, which was entropy change for the protein. So it was unfavorable because these proteins are tightly packed. So it was positive and it was unfavorable. Whereas T delta S of solvent, so now water was freely moving in the protein crystal. So it was highly favorable, range from minus 7.5 to minus 50 kilocalories per mole. So if we combine all these terms and put it into the delta G of crystal, then we will see that positive means delta H of crystal was negative and small, whereas minus T delta S protein was large, but it was positive, whereas T delta S solvent was large, but it was negative. So these larger terms will cancel out the effect of each other, and ultimately the delta G of crystal was negative, but it was small, so which means that it is slightly negative, so crystal formation will take place. So delta G, if it is negative, crystal formation is means the formation of crystal is favorable. So this is how the thermodynamics of crystal growth is studied, and this shows that why the formation of crystal is favorable, and it is favored means one of the main factor which is responsible is the T delta S of solvent, which was highly negative. So this is the factor which is means dealing with the effect of T delta S of protein, because it was unfavorable. So it is cancelling out the effect. That's why the entropy change brought about by the solvent is one of the deciding factor that why the delta G of crystal is favorable, why it is negative. Now we will talk about the strategies to improve the protein crystallization. So till now we have seen that how protein crystallization takes place, why it is important, and now we will talk that how we can improve the protein crystallization. So main thing is to reduce the entropic penalty. So in the previous thing we have seen that this particular T delta S protein term, it is acting as a liability for us because it is highly unfavorable. So how we can deal with this? So now in this we will try to reduce this entropy penalty, which is of T delta S protein. So first thing which we can do is cleaving the histone tags and flexible regions. So as we have seen that during purification, when we were performing affinity chromatography, so that time to obtain our protein of interest, we have attached histone tags to it. So now to reduce this entropy term, means to do this, we cleave those histone tags and we also eliminate the flexible regions. So there are certain regions which are disordered and they are very flexible. So we need to eliminate those floppy termini and we need to remove those regions. Okay, so we need to remove these floppy termini so that we can reduce the entropic penalty. Second thing which we can do is expressing the individual domains separately. So in these cases where we have a bigger protein molecule, so instead of expressing its both subunit at the same time, what we can do is that we avoid the unstable multi-domain interactions. We can break it off and we can use the individual domains and then observing their structure individually instead of observing entire bigger protein complex at the same time. So this we try to avoid the multi-domain interactions which might interfere or which are giving the rise to which is increasing the T delta S protein. And next thing which we can do is adding ligands or binding partners. So which stabilize the protein structure. So in this way we can add certain ligands so that if we have two different domains or if we have a larger protein, then those ligands they bind to it and they will stabilize its structure so that proper crystal formation can take place and we can reduce this entropic penalty. Fourth thing which we can do is mutating the high entropy residues. So there are certain residues such as glutamine or lysine. So we can replace them with alanine or serine so that they can reduce the entropy. So in this way, they will reduce the conformational flexibility and ultimately they will reduce the entropic penalty of the protein. So in this way, these are some of the methods or some of the strategies which generally different research groups use and they have personalized them according to their needs. So which helps them to compete with the large scale structural genomic initiatives. So in this way, these strategies will help you to get the proper ordered crystals and the formation of the crystal will be favorable. So in order to summarize all of this, the protein crystallization, it is essential for X-ray crystallography, but it is still a trial and error based method. So till now, as we have seen that there is no well-defined or standard concentrations or standard way in which proper protein crystallization can be achieved. So it is still a hit and trial method, so trial and error method. And we have also understood that crystallization is the major barrier towards achieving success. So according to the data published by JCSG, so in this they have shown that we have the target protein sequences in which we have selected targets of about 33,000 and after those obtaining the selected targets, we have cloned them. So the success rate which was obtained was the 84%. And after the clone targets which are obtained, we allow them to express because these are the genes. Now in expression, they will be converting into protein. So expression of 27,959 when they were cloned, when they were undergone expression. So 27,640 were overexpressed. So there is the 99% success rate from cloning to expression. But if we move from these expressed protein to crystallized proteins, then there is only 8% success. So which shows that this is a bottleneck effect, that how it is limiting the success of these protein identification or the means this X-ray crystallography. So it is hindering because we are not able to crystallize maximum or majority of the proteins. So this shows that crystallization is the major barrier. And out of these crystallized structures, the solved one are 1045, which is about 49% success. And then from these solved, the structures which are deposited in PDB, so there is 93% success rate. So by looking at this data, we are clearly able to see that the crystallization is the major barrier. So what we want to do, we want to know that how proper crystallization can be obtained, whether we can design certain strategies which will help us to make proper protein crystals. So our main aim is to study that. That's why SGRC and relative initiatives, they are focusing on solving the 3D protein structures for health and biotech applications. And then crystal diffraction amplifies the weak molecular signals, but perfect ordering is required. So this perfect ordered crystal is also one of the prerequisite to observe proper diffraction patterns. Thermodynamics governs the crystal formation. So as we have seen in delta G equation, that how it governs that whether you will be able to obtain proper crystal or not. So with solvent entropy playing a major stabilizing role. So solvent entropy was highly negative. That's why we are able to observe the proper protein crystal. Then various strategies exist to optimize the crystallization, which we have already seen, that we can remove the flexible residues, or we can study the multi-domain protein and we can study its individual domains separately. We have seen that we can replace certain residues with less volatile or means the residues which were previously such as glutamine or other residue, we can replace them with some alanine or other residues. So these are some of the strategies which are used. So this is all about the crystallization and how the crystals are formed. So we have covered it. Now next main step is the crystal mounting. So now till now what we have seen so far, that we have seen that we have the gene of interest, we first clone it, we express it to obtain the sufficient quantity, then we undergo PCR amplifications and other rounds. We allow it to express in the host so that a sufficient quantity of protein is obtained. Next step was to purify that protein so that we can get the homogeneous protein concentration, because our next aim was to make the crystals. So for crystal formation, we require the homogeneous mixture of proteins and sufficient quantity of the protein. So after the purification is completed, next step is the crystal formation. So now when the crystals are properly formed, the next step is to mount them so that we can pass the X-ray through them. So crystal mounting is also a very important step. It ensures that protein crystals, they are properly positioned for X-ray diffraction. So if they are not positioned properly, then also it will interfere with the X-ray diffraction pattern. So the challenges with this particular problem, means this crystal mounting, is that these protein crystals, they are very fragile. They can break easily. So for example, if you have, means this protein crystal formation is also a time consuming process. Now imagine you are means getting the protein crystal after waiting for many months, and then when you are means performing the mounting of crystal and then it breaks, then you can see how much pain you will feel, that you have put your so much hard work into crystal formation and now you lost that crystal. So that's why the challenge in this mounting is that protein crystals are very fragile. So you means they can break easily. So and they must remain in contact with the mother liquor so that you can prevent the dehydration, because we have seen that about 30% is the this solute only, which is sorry, 30% is the other concentration which is present inside the protein. That is the solvent concentration. So we want it to be in the contact with the mother liquor so that there is no dehydration, because dehydration will also break our crystal. So these are some of the challenges. And in this, I have shown that how these crystals, means how they are mounted and how they appear when we take them and mount them. So this is also the technique. Then in the next slide, I'll discuss that how with the loop we pick up the crystal carefully, we perform its, means we allow its, we cryopreserve it, and then after cryopreservation, we use anti-freezing treatment when we want to mount it. So flash cooling and cold nitrogen gas is passed through it, and ultimately, after all these things have been done, now we will allow the X-ray to pass through it. So means now next slides we will see that why we want to cryopreserve it, why we need it, why we need cold treatment. So before moving that, we firstly need to identify among the protein crystals. So now you have performed the crystallization, but before mounting the crystal, you want to make sure that the crystal which you are picking up, that is a protein crystal and not some impurity, or it is not a salt crystal. So there are different methods which exist. One such method is protein dye staining. So protein dye staining means you will use some dye which will stain your crystals. So now protein crystals have the ability to get stained with the specific dye. Only they will get colored. and salt crystals won't take up the dye. So in that way, you will be able to distinguish that whether the crystal which you are selecting is a protein crystal or the salt crystal. So staining method is one. Next method is the crushing test. So protein crystals, they crush easily into the smaller fragments, whereas salt crystals, they crush into fewer and larger pieces. So this is also one of the tests which you can perform to distinguish that whether the crystal is protein crystal or is it a salt crystal. Next test is the dehydration test. So as we know that these protein crystals, they are composed mainly of they means have a lot of water, a lot of solvent inside them. So they will disintegrate as soon as they get dehydrated. Whereas salt crystals, they still remain intact. So this is also one of the method with which you can distinguish that whether the crystal is the protein crystal or the salt crystal. Fourth such test is the X-ray diffraction test, which is the final confirmation. So protein crystals, they will show the characteristic diffraction patterns, whereas if you do not have protein crystal, then you won't be able to achieve different diffraction patterns. So these are some of the methods which you can identify that the crystal which you have obtained is the protein crystal. Now we will talk about different tools which we generally use to mount these crystals for X-ray diffraction. So protein crystals, they are mounted using the cryo loops. So in this case, this is the forceps with the cryo vial. And then there are specific tools like tong. So these are the tools which are helpful to pick up these loops. So these means the loops, they have, why they are called loop, because in their terminal they fold like a loop. So these loopy like structure, they will be able to pick up the crystals. So you have to carefully pick up the crystal from this, and these particular means these instruments, or you can say these loops, they are attached to the specialized tools such as tong. So in this case, you can see how this loop is attached to the tong. So this tong will help you to pick up the crystal. You will not directly use these loops. So you will use either tong or the forceps. So with forceps, you can pick the cryo vial, and then you can take the tong with the loop, and then you can pick carefully the crystal. Or you can also use the wands. And then if we talk about cryo loops, then cryo loops are also means they also come in variable sizes depending on that what is the size of the protein crystal. So depending on that, you can use any of the cryo loops. So they are different variants such as red, green, yellow, blue, and larger sizes. So this is the means their size limit. So this has the red one has the limit of 0.025 to 0.05 millimeters. Similarly, green, which has a slightly bigger size, so 0.05 to zero point 0.1 millimeter. Then yellow one is a little bigger from the green one, which has the size of zero point 0.1 to zero point 0.2 mm. Then blue one has zero point 0.2 to zero point 0.3 mm, and then means larger sizes up to 1 mm, so which are available for the larger crystals. So depending on that what crystals you have, you can select any of these loops which are means desired or which can pick up that particular crystal, and these colors will help you to means distinguish among different loops. Then what are the steps for mounting the crystals? So first step is to pick up the crystal using the loop, and you have to make sure that you do not means this should prevent the dehydration, because in the previous slide we have seen that how dehydration will destroy the protein crystal. So you need to prevent dehydration, and then you have to transfer it to the cryo vial for freezing. So in this case, this cryo vial, you have to properly place your um, the crystal which you have picked with the loop. So you have to properly place it into the cryo vial, and then for freezing, and then you have to mount it onto the goniometer for precise positioning. So positioning is also very much important. Okay, so means how you position it onto the goniometer is also very much important, because it will also determine that whether you will be able to observe proper diffraction pattern or not. So these are some of the steps. Now we will talk about that why do we need cryopreservation of crystals. So why we freeze them? So to prevent X-ray damage to the crystal, because we have seen that these X-rays, they have high energy and they can destroy the crystal. So to prevent it, we need to freeze these crystals, and this freezing, it also helps to maintain the structural integrity during the data collection. So to means maintain the structural integrity, we need to freeze the crystals. Now, how to freeze these crystals? How the freezing is done? So first thing is flash freezing. So in flash freezing, what we do? We take in the liquid propane or freon. So we put our crystal into these liquids and they will quickly freeze it. So this is known as flash freezing in minus 70 degrees centigrade. Next step is, or next way with which we can freeze it is the nitrogen gas freezing. This is also done at minus 70 degrees centigrade. So this is done to prevent the hexagonal ice formation. So to avoid the ice formation, we can also go for the nitrogen gas freezing. And then ultimately there are certain cryoprotectants which already are present, such as polyols, salts, sugars, and solvents. So they can also be used for freezing. So in this way, these are different methods with which we can freeze our crystals. And why freezing is done? To avoid the X-ray damage and to maintain the crystal in proper structural, to maintain their structural integrity. Now we will talk about the goniometer, because on the top of it only we are mounting the crystal. So goniometer is responsible for the crystal alignment for data collection. So goniometer is used to precisely adjust the position of the crystal in the X-ray beam. And there is three circle goniometer which allows two type of rotation. So this is the goniometer in which you have placed your crystal, protein crystal. So it allows three different circular, means two types of rotation, and there are three circles. So first rotation is the omega rotation, which moves perpendicular to the X-ray beam. So in this case, if your X-ray beam is coming in this way, so the axis which is perpendicular towards the X-ray beam, that will undergo omega rotation. So this is the omega rotation. This is axis is the perpendicular axis, and this rotation is known as omega rotation. Then comes phi rotation, which rotates around the crystal axis. So this is the axis of crystal, and this is which is rotating around the crystal axis. This is phi rotation, and the rotation which is perpendicular to the X-ray beam is the omega rotation. And there is a third fixed angle which is about 54.74, which helps to optimize the diffraction collection. So one angle is fixed. So one angle is fixed, then another is omega and phi angles which are continuously varying. That's why we allow two types of rotation. This angle is fixed. So in this way, we are rotating the crystal to obtain proper diffraction pattern. And we align the crystal in the X-ray beam. So rotate omega and phi axis to check the movement, and we need to adjust the goniometer screws until the crystal remains centered. Align the crystal lattice by centering first order diffraction layers. So why we are using it? So that we can properly align our crystal lattice so that it is in the centered, so that proper X-ray can pass through it and proper diffraction pattern could be achieved. Now there are certain issues with the crystal diffraction quality. So now what can happen is that if there is some disorder while doing all of this, there might arise some rotational disorder. So as I've told you earlier also, that there are different unit cell layers which are overlapping or which are present onto each other. So if there is some means disorder among those layers, there is some rotational disorder, then there is misalignment of molecules which will reduce the diffraction clarity. Then translational disorder. So if there is translational disorder, then also there will be random shifts in crystal lattice which will degrade the diffraction quality. So you need to make sure that your crystal, it should be in perfect order to obtain proper diffraction pattern. So these are the issues in the crystal lattice. Then comes diffraction patterns and data collection. So if there is symmetry in the diffraction spots, so which help us to determine the unit cell dimensions. If there will be symmetry, then you will be better able to distinguish among the atomic positions, and you will be able to determine the unit cell dimensions. Spacing between the spots, it provides the information on the crystal lattice. And higher order diffraction layers indicate precise atomic structures. So the diffraction layers, if they are highly ordered, then you will be better able to obtain the precise atomic structures. So this shows that there are different um, issues which are involved in the crystallization, and means there should be proper order in the crystals. So if the crystals are not properly ordered, if the layers are not properly ordered, then you won't be able to get proper diffraction pattern. So symmetry is very important to obtain the proper atomic resolution diffraction patterns. Now a little bit of historical background of X-ray crystallography. So X-rays, they were discovered by Wilhelm Röntgen in 1895, and in 1901 he was awarded the first Nobel Prize in physics. So in 1901 the Nobel Prize has been started, so he was the one to obtain the first Nobel Prize in physics. And in 1914 Darwin's formula established the relationship between the diffraction intensity and the unit cell. And if we talk about the present day, then the X-ray crystallography is the primary method for determining the protein structures. So this is the primary experimental method which is considered to be very confident, or we can say which gives the better means proper high resolution protein structures. So that's why this method is still the primary method. Now we will practice few MCQs, but onto this I also wanted to add that although there is the technological advancements, there has been upcoming of the machine learning and the artificial intelligence methods, but still X-ray crystallography is the primary method. It is the experimental method. So it is already verified, or you can say it is the validated. So the structure which is visualized by the X-ray crystallography, that is the validated structure. Whereas we already have seen that how AlphaFold has come up and it has revolutionized the field of structural biology. So it is based on the already available knowledge and with which it predicts the protein structures. But still it has certain limitations. Whereas X-ray crystallography is means if it gives certain structure, so it is expected, it is validated structure. So that's why these methods are very important. Although it has a lot of limitations, there are many difficulties to obtain the proper structure, but still this method is the main method, or you can say this is the ground truth. Now we will discuss certain MCQs which were present or which were there in the previous uh, means in the previous course, last year course. So these MCQs will help you to understand better that whatever you have learned till now, whether you are able to follow it up or not. So now let's begin with the first practice MCQ. So which of the following is a low resolution structural biology technique? Option A, single angle X-ray diffraction. Option B, cryo-EM. Option C, NMR. And option D, all of these. So now you have to tell that among all of these, which one is the low resolution structural biology technique? Okay, so I'll give you some seconds and then I'll discuss the result. Okay, now let's see. So the right answer is small angle X-ray diffraction. So we have already discussed that the cryo-EM and NMR, they are the high resolution structural biology techniques, as well as X-ray diffraction is also high resolution, but the small angle X-ray diffraction, it is the low resolution technique. So this is just the representation that when the X-ray is coming, it is passing through the crystal. Then if this is known as wide angle X-ray diffraction, whereas this is diffracting at the small angle. So small angle X-ray scattering, or it is also known as SAX. So it is a low resolution technique which is used to study the overall shape and size of the macromolecules in solution, unlike cryo-EM and NMR, which provide high resolution structural details. So that's why the right answer is option A. Now let's move to question number second. So now second question is doubling time for E. coli is option A is 40 minutes, option B is 20 minutes, option C is 10 minutes, and option D is 60 minutes. So now you have to tell that what is the doubling time for E. coli. Okay, now let's see. So the right answer is option D, which is 60 minutes. So doubling time of E. coli depends on the growth conditions. Now you might think that you might have seen in certain literature that E. coli's doubling time is 20 minutes. So now I will tell you that why is it so. So if there are optimal conditions, which means if you have a rich medium and temperature is 37 degrees centigrades, then in such optimal conditions, the doubling time is 20 minutes. But if there is a minimal medium or nutrient limited conditions, then this doubling time is 60 minutes. So if the if it's 60 minutes, then that means there is a minimal medium or nutrient limited conditions. So in this way, do not get confused. It depends that what is the medium condition. So this is the worst case scenario, 60 minutes. But in best case scenario, the doubling time is 20 minutes. Now let's look into the next question. So the next question is, purification of proteins using the nickel NTA column is an example of option A, SEC, which means size exclusion chromatography. Option B, ion exchange chromatography. Option C, hydrophobic interaction chromatography. And option D, affinity chromatography. So now you have to tell that this purification using the NiNTA column, it is the example of which one of these techniques. Okay, now let's see. So what is the right answer? So the correct answer is the affinity chromatography. And we have also seen in the previous slides that why is it affinity chromatography? Because there is some kind of binding which is happening in this case. So what happens in nickel NTA column in this particular affinity chromatography? We have the column in which we already have the NTA, which is a chelator, and the nickel ion, which is attached to this NTA. So this is the nickel ion which is attached. Now when we add the protein mixture in the column, then for the desired protein of interest, we have the histidine tags, which are attached to this protein. Now the proteins which have these histidine tags, they will interact with these solid beads which are attached to NTA as well as nickel. So they will interact with it. And in these histidine, there are imidazole rings which are present. So their nitrogen are interacting with the nickel molecule, or you can say with the nickel ion. So with this interaction, the proteins which contain the histidine tag, they won't be able to elute out. They will not pass through the column. They will get bind to these beads. Whereas other proteins they will completely get eluted out. So ultimately, in the end, we add imidazole, which will replace these histidine tagged proteins. Imidazole will bind to nickel, and now the protein with the histidine tag will be eluted out in the end. So this is one of the way when we were studying protein purification, then we have seen the affinity chromatography and we have seen that how the binding is happening and why it is called affinity chromatography. Whereas if we talk about size exclusion chromatography, so in this case, the separation is happening on the basis of size. There is no such binding which is happening. In this case, the pores are present and those pores, small pores are present in the gels, and then the proteins which are smaller in size, they will get trapped in those pores, whereas the proteins which are larger in size, they will elute out first. So in size exclusion chromatography, the larger proteins elute out, and then in the end, smaller proteins get elute out. For example, if we talk about ion exchange chromatography, then in this case, as the name suggests, ion exchange. That means in this case, separation is happening on the basis of charge. So in this also, we have the column in which on the surface of the column, we either have a cation or the anion. And depending on the charge present on the surface, the oppositely charged protein will bind to it, whereas the other protein get eluted out. So this is the ionic exchange chromatography. And then comes hydrophobic interaction chromatography. So in this case, there will be some hydrophobic interactions. So the proteins which contain those alkyl groups or the groups which can be involved in the hydrophobic interactions or the hydrophobic amino acids, they will be interacting with the surface, whereas the other proteins which lack these hydrophobic groups, they will elute out. So these are different protein purification techniques, and NINTA is among the affinity chromatography technique. So I hope it will be clear till now. Now let's look into the fourth question. So the question is, which of the following technique separate the proteins on the basis of their size? So this we have just discussed. So I hope you will be able to answer it. So option A is ion exchange chromatography. Option B, affinity chromatography. Option C, gel filtration. And option D, chiral chromatography. So now you have to tell that which of the following techniques separates the protein on the basis of their size. So I will give you 10 seconds and then we will discuss its answer. Okay, so now let's see. So the right answer is option C, which is gel filtration. So gel filtration is also known as size exclusion chromatography or gel permeation chromatography. So these are three different names. So do not get confused. And even if you do not able to identify that whether it is size exclusion, then you can simply cancel out. You can use the technique of cancelling out the other options. So as it is clear that ion exchange is the separation on the basis of charge, affinity is on the separation of binding site or binding interactions, and chiral chromatography is in some other technique which is not on the basis of size. So in this case, the technique which is left is gel filtration. Now let's move to next question. So this question is identify the equation stated below. So the equation is 2d sine theta is equals to n lambda. Options are Michaelis-Menten equation, option B Bragg's equation, option C Fourier law, and option D Darcy equation. So this equation was also discussed, not in detail, but a brief discussion happened in the week 3 classes. So I hope you will be able to recall it. Okay, now let's see. So what is the right option? So the right answer is option B, which is Bragg's equation. So this is the Bragg's equation in which it has shown that for a constructive interference, n lambda should be equals to 2d sine theta, which is Bragg's law, where d is the space between these two unit cell layers. So d is the distance. And then this is the incident plane wave, and this is the wave which is getting diffracted. So this is the angle which is 2 theta, and this is the theta with respect to the plane, the angle which is made. So in this way, 2d sine theta is equals to n lambda. So this is the Bragg's equation. If we talk about Michaelis-Menten equation, then this is involved in the enzyme kinetics. This is not there. Then comes Fourier law. So this is also not involved. Darcy equation. These are different equations. So the right answer was Bragg's equation. And in the later sessions or in the later weeks, we will also look into the detail. So at that time, we will discuss that how the means what each and every term means and how they are linked and how they decide that whether you will be able to achieve the constructive interference or non-constructive, or you can say destructive interference. So constructive interference is very important so that you can properly visualize the molecules or atoms. Atomic level resolution will be better if there is constructive interference. So now let's move to next question. So this is question number 6. In the following equation below, the term delta H is. So now you have to tell that what is delta H, whether it is favorable or non-favorable. So this we have already discussed when we were talking about the thermodynamics of crystal formation. So delta G of crystal is equals to delta H of crystal minus T delta S of protein plus delta S of solvent. So you have to tell what delta H term is. Option A, it is highly favorable, that is minus 100 kilocalories per mole. Option B, it is unfavorable. Option C, it is moderately favorable, that means 0 to minus 17 kilocalories per mole. And option D is none of these. So you have to tell what delta H crystal term will be. Okay, so now let's see. So what will be the right answer? I hope you have guessed the right thing. So the right answer is option C, which is moderately favorable. So when we were discussing about it, so then we also talked about the value. So it ranges from 0 to minus 17 kilocalories per mole, and it is not minus 100. So when we were discussing it, then we discussed that the delta H crystal is moderately favorable, whereas T delta S of protein was highly positive, which is unfavorable, whereas T delta S of solvent was highly negative, which means it was highly favorable. So in that way, this T delta S of solvent was the bigger term, which was helping us out, whereas T delta S protein, it was a liability, it was the term which was means decreasing, or which was means interfering, which was highly unfavorable. So that's why delta H of crystal is moderately favorable. It's not that high if we compare it with the T delta S of solvent. Now the next question is, 0 degrees of orientation and translations are found in option A, protein in water, option B, protein crystal, option C, protein in buffer, and option D, none of these. So now you have to tell that where we find 0 degree of orientation. That means 0 degree of freedom in orientation and in translation. Okay, so now let's see that what is the right answer. So the right answer is option B, which is protein crystal. Now we'll see that why protein crystal. So if protein is present in solution, then you can see as protein in water. So it is same as protein in solution. So now if protein is present in solution, that means there is these proteins are present far away from each other. So when they are lying far away from each other, then there is no such lattice contacts which are taking place. So they are free to move. So that's why they have 3 degrees of freedom in orientation as well as translation. Whereas if we talk about protein crystals, then these individual protein units, they are closely packed, they are compact, they are in the lattice, there are lattice contacts between these units. So now there will be 0 degrees of freedom in orientation. They are not free to move. So their entropy is means less. They are not free to move. There is lesser noise as compared to protein in solution. So 0 degree of freedom of orientation and translation is found in protein crystal, not in water, not in buffer, and none of these will also not be right because the right option is protein crystal. So I hope you got the idea that why it is 0 degree of orientation and translation in protein crystals. So now let's move to next question. So in this equation, in the equation below, the term T delta S solvent for protein crystallization is. This is the equation, delta G of crystal equals to delta H of crystal minus T delta S of protein plus delta S of solvent. So you have to tell this particular term, that is T delta S of solvent, whether it is favorable, it is unfavorable, it is neutral, or none of these. So what do you think? What will be this term? So I hope by now you have got this equation into your head. You will be better able to distinguish. I hope now it will be more clarified. Okay, now let's discuss the right answer. So the right answer is favorable. And I will also tell you why it is favorable. So when we talk about T delta S of solvent, so in this case, we are talking about the entropy of the solvent and not of the protein. When we talk about delta S of protein, in that case, it was highly unfavorable. But if we talk about delta S of solvent, then it is highly favorable because when protein is present in solution, then these water molecules, they are not free to move because they're attached to protein and only protein is moving, not the solvent is moving. So in this case, there is 0 degree of freedom in orientation and 0 degree of freedom in translation for the solvent, or you can say for the water molecules. Whereas if we talk about protein crystals, then in this case, each protein unit that is tightly in the crystal interaction, or you can say in the crystal lattice, there are lattice interactions between these protein crystals. These units are tightly packed. So in this case, the water is not able to bind to these proteins. So water is free, or you can say solvent is free to move. So entropy of solvent in the protein crystal will be very, very high. So that's why it has 3 degrees of freedom in orientation and 3 degrees of freedom in translation. So it is favorable. Okay, so now let's move to the next question. So the next question is, which of the following can be used to distinguish or differentiate between a protein and a salt crystal? Option A, protein dye test. Option B, crushing the crystal. Option C, dehydration. And option D, all of these. So this is also a very easy question. So I'll give you some time to answer this question. Okay, now let's see what is the right answer. So the right answer is option D, which is all of these. So when we were talking about distinguishing between the protein and salt crystals, then we have seen that there are protein dye tests. So protein dye test, what they do, they color the crystals which are protein crystals. And these protein crystals are the ones which will take up the dye and which will get colored. The salt crystals are not able to take up the dye or show any color. So this is one of the way with which you can distinguish among the protein and the salt crystals. The second thing is crushing the crystal. So at that time we also discussed that when you crush the protein crystal, it is very fragile. It will break into smaller units, multiple smaller units. Whereas if you crush the salt crystal, it will break, but it will break into the larger units. So the units which are obtained, they are a little bit large. They're not very small or fine. And then the third thing is dehydration. So this is also one of the tests, because when we were talking about the protein crystals, then we talked that in these protein crystals, it contains a lot of water inside it. So if it get dehydrated, then it will get break. Then it will be broken into smaller units, whereas salt crystals, they do not get affected by dehydration. So this is also one of the distinguishing technique or distinguishing test for the protein and salt crystal discrimination. So that's why the right option was all of these. Now let's look at the 10th question, which is the last question. So identify the method of protein crystallization given in the image. So this is one of the method for protein crystallization. So this comes under vapor diffusion methods. So we have not gone into the detail of these methods because they have already been discussed in detail in your week 3 classes. So in this, we have the apparatus in which there is a precipitant solution. This is the grease which is attached, and then the cover slip which is put on it, and then it contains the protein plus the this is the precipitant solution. So in this case, you have to tell which method is it. So option A is sitting drop vapor diffusion method. Option B is under oil micro batch crystallization. Option C, hanging drop vapor diffusion method. And option D is dialysis. So this is also quite straightforward. Okay, so I'll give you some time to think and then we'll discuss the right answer. Okay, so now let's talk about the right answer. So the right option is hanging drop vapor diffusion method. So one of the means clear thing with which you can identify which method is it. So in this case, this particular protein drop, it is hanging. So that's why it is a hanging drop vapor diffusion method. Whereas in case of sitting drop, you will see that in this case, instead of this cover slip containing the protein, you will see that the protein will be kept somewhere here. So it will not be hanging. So in that case, it is known as sitting drop vapor method. Whereas in under oil microbatch crystallization also, the drop will not be hanging like this. So it will also be in a proper orientation, the protein will be present. with the oil. And then in dialysis, so it is like a membrane in between with which the movement of the protein and the precipitate solution will happen. So this is also not the right option. So the right option was hanging drop vapor diffusion method. So I hope the things which we have discussed today are clear to you. And this is all about today's session. So if you have any doubts, then you can either, if you are watching this YouTube video, then you can comment on the video and you can ask your doubts, or otherwise you can also ask your queries in the discussion forums. So in that also I'll try to answer it. And if you have any doubts about the certain material which was not covered in this, or if there are certain topics for which you want some study material, then also you can comment or you can ask me directly. So I will be happy to help you. So I hope you have got a lot of idea about today's session, about all those topics which were covered. So I'll end the session on this note and I'll meet you in the next session. Till now, goodbye, take care.